Hello, everyone. Welcome to today's edition of Pathcast. Today is September 3rd, 2024. It's a Tuesday, and I'm Rifat Mandan in California. And today, we are very delighted to have with us Dr. Robert Ohgami, who is currently professor at the University of Utah School of Medicine. And he also serves as the Vice President of Research and Innovations at the ARUP Laboratories. And he's going to deliver our continuing series on hematopathology. And in fact, this would be the 10th lecture in the series. And today he's going to talk on spotlight on innovation, practical advances in rare and orphan hematologic diseases. And as many of you are probably aware, that today's lecture is accredited by City of Hope for 1 CME credit. And to access the CME credits, and in fact, both international and those audience or who are listening to it from within the United States, they can access the CME credits. For that, they have to register to the City of Hope CME website, which I will post in the chat box soon. And if you are watching from inside the United States, you can just text the code to this phone number. And for the international participants, they can email the code to this email address that you see on the screen, that is cme@coh.org. And please remember that the attendance is recorded only during the live lecture. And once the lecture is over and you send the CME code, so you will get an evaluation from City of Hope. And please submit your feedback within 1 business day so that you can get the CME code. Without much ado, I would like to hand over the microphone to Dr. Ohgami. And thank you so much, Dr. Ohgami, for joining us today. Over to you now.
Thank you for that introduction, Dr. Manan. Let me share my screen. All right. Great. And I'm also going to focus in on my presentation now. So thank you for the opportunity to speak today. My talk is entitled Spotlight on Innovation, Practical Advances in Rare and Orphan Hematologic Diseases. So you may wonder this talk is about. So first, a little bit of paperwork. Disclosures, I don't have any relevant financial relationships. And then also here. So getting back into our talk now. Again, spotlight on innovation, practical advances in rare and orphan hematologic diseases. We're going to be talking about the following. So what are the goals for this talk? The goals are to discuss practical updates in several rare and orphan diseases in hematopathology, to discuss newer and emerging technologies in hematopathology and hematology, including artificial intelligence, and really thinking about what technologies we want to watch out for. So the outline for this talk is as follows. There's an introduction where I will cover a brief history of hematopathology, given background on what are rare and orphan types of diseases in general. And then there's a body to the talk where we're going to review several rare and orphan diseases. And obviously we can't cover every single rare and orphan disease in hematopathology, but there are 3 key ones that we can use to understand the impact of technology and potential future applications of technology in different types of rare and orphan diseases. And those rare and orphan diseases that we're going to cover are Blastic, Plasmacytoid, Dritic cell Neoplasms, Castleman disease, and pediatric type follicular lymphoma. As we go along discussing these rare and orphan diseases, we'll interlace current technologies, how they're being used, and future types of innovative technologies and their application in these diseases, as far as diagnostics and prognostics. And then we're going to spend a fair bit of time talking about artificial intelligence, which really is a part of the current now and also the future. With that, let's get in on our talk here as far as discussing a little bit of brief history of hematopathology classifications. So in the 1860s, we started out with a classification system by Virchow and Cohnheim, which had an approach to hematopathology using morphology, the microscope, identifying disease entities like lymphosarcoma. In the 1920s, Oberlin, Brill, and Simmers had a classification system identifying different types of lymphomas as reticulum cell sarcoma or giant follicular lymphoma. Again, largely based on morphology and microscopy. In the 1950s and 60s, we get the Rappaport, Winters, and Hicks classification, again, utilizing microscopy and morphology, identifying different types of diseases based on architecture and cell size. And then in the 70s, we get the Lukes and Collins, Keel, and British National Lymphoma Investigation and NCI working formulations, which use not only morphology, as well as also clinical information and background on patients, but also immunophenotyping. We start to see the integration of immunophenotyping. In 1994, we get the real classification, where we again are using morphology, immunophenotypic information, cytogenetics, and we start to see the emergence of molecular as far as clonality, T cell clonality and B cell clonality. And then in 2001 to 2017, we see the WHO classification, where there's an era of, again, looking at morphology, immunophenotype, cytogenetics, clonality, and also molecular. And then in 2002, we see the emergence of two classification systems, the WHO classification and ICC, which integrate multiple different types of ancillary and primary data, morphology, immunophenotype, molecular, and cytogenetics. And as you see, as you go along in time, the way that our classification systems have been progressing or have been really following the emergence of technologies, new technologies. Each time a new technology is released into the world, you see that our hematopathology classification eventually closely integrates those new technologies and utilizes them for the best diagnostic care, from molecular, with DNA being discovered, to one color flow cytometry in the 1960s. And then you see, again, with the NCI working formulation, et cetera, that you start to see years later the integration of immunophenotyping, Sanger sequencing in the 1970s. And again, as flow cytometry becomes more and more multidimensional, you see the importance of that broader kind of denser data over time. The integration of things like nanopore technology, PacBio, and again, the emphasis on molecular. You'll see what we see in the hematopathology classification systems are that we are integrating more and more molecular phenotyping over time. And so the point of this, just to distill it down, is that technology really drives medicine. And this is why we need to follow and understand new technologies. So that said, we have a little bit of background on why it's important to understand innovation and technology in our hematopathology classifications. But again, this talk is also about rare and orphan diseases. And what are rare and orphan diseases? So the definition of a rare and orphan diseases is essentially a disease that impacts a small number of people. And that percentage varies depending on whether you're in the United States or in Europe, but in general, it's less than 1 in 200,000 to 2,000 persons. The thing about rare and orphan diseases is, unfortunately, since there are fewer number of individuals impacted, these tend to be under-researched and underfunded, and therefore there are fewer treatment options available. So some examples of these, and we're going to cover this, is Castleman disease, Blastic plasmacytoid dendritic cell neoplasm, and pediatric type follicular lymphoma. These are rarely seen compared to a lot more other diseases that are more common, such as iron deficiency anemia. So these I called non-examples. 10% of the US population or more actually have this. Acute myeloid leukemia, 10 in 200,000 people have acute myeloid leukemia. So where are we now in the talk? So we've gone through our introduction, and now we're into our body, and we're going to start to talk about blastic plasmacytoid dendritic cell neoplasm. And I'll give practical diagnostic pearls, as well as understanding, again, how technology impacts or may impact our diagnostic system for blastic plasmacytoid dendritic cell neoplasms. So bpdcn, just to do an overview here and understand it. The definition of bpdcn, it is a rare and orphan disease, as I said before. It impacts approximately 1 to 5 patients per 1 million people. And it's a malignancy of plasmacytoid dendritic cells. Typically, patients will present with skin involvement, lymphadenopathy, and then also systemic dissemination is frequent, including bone marrow involvement. It is critical to understand blastic plasmacytoid dendritic cell neoplasm, even though it is a rare and orphan disease, because the therapies differ, and it needs to be distinguished from acute myeloid leukemia, or AML, as well as mature plasmacytoid dendritic cell proliferations that can be seen in CMML and then also AML. And so it can be tricky to diagnose. And so how do we diagnose bpdcn, blastic plasmacytoid dendritic cell neoplasm? Well, histopathologically and cytologically, what you will see is medium-sized cells with eccentric nuclei, a plasmacytoid kind of appearance, that are agranular and will have vacuoles in the cytoplasm, as shown in the figures on the right. In the skin, a skin biopsy will show dermal involvement, but generally not epidermis involvement at the surface. Bone marrow involvement by this disease may be subtle or actually quite diffuse, and so it can be a bit tricky to identify. bpdcn has a characteristic immunophenotype. The classic immunophenotype is it is a CD123 positive, CD4 and CD56 positive neoplasm. So 1, 2, 3, 4, 5, 6 positive neoplasm. But there are cases that can lack either 123, CD4, or CD56. And then additional plasmacytoid dendritic cell markers are useful in being able to actually call something bpdcn, such as TCL1, TCF4, CD303, or 304. What's key with bpdcn is that flow cytometry is really important in many cases. It helps to have that flow cytometry multi-dimensional data to be able to actually call bpdcn versus other types of neoplasms like AML. And so, again, what kind of technologies play a key role in diagnosing bpdcn? So microscopy, it is a type of technology, albeit older. Histopathology, flow cytometry, and immunophenotyping is absolutely essential. And what is flow cytometry, or how do we utilize flow cytometry today, this technology? Well, there's classic flow cytometry, if you want to think of it this way, where you have antibodies that are directed against different antigens on the cell, on cells of interest, and are labeled with different fluorophores. We detect the antigen expression via these antibodies that are labeled with different fluorophores, like green fluorophores, essentially, to say that there is CD8 expression on the cell or something like CD57 expression. Lasers and detectors are an important part of flow cytometers, enabling us to, again, identify the expression of these different markers. Classic, what I'm calling classic flow cytometry, requires understanding or interrogating and distinguishing between different color channels. And there can be issues there with as far as spillover or overlap of different colorimetric spectra, which can affect the ability to identify different proteins on different cells accurately. But over time, this technology has advanced, and initially there was one protein that was able to be identified essentially by flow cytometry. Over time, we became better and able to distinguish 2, then 4, then 10 different types of proteins and fluorophores. And now there are newer technologies that exist. Technologies like spectral flow cytometry. This is a technology to watch out for or to be aware of, and will gain more use, where spectra become a parameter, so that we're able to detect essentially 40 colors in one tube. And this is the key here. By looking at a fingerprint of a different emission from a particular fluorophore. Spectral flow cytometry gives us this really dense, multidimensional data that you can see here, and it allows us to pull out many, many different cell types. These are different types of visual depictions of this high dimensional space using different types of approaches like Visney or Tisney plots, because it is hard for us as humans to look at such high dimensional space. And so why do we pay attention to this kind of high dimensional flow cytometry, and how is it impactful or going to be impactful in the future? Well, this gives us great efficiency gains. In flow cytometry, instead of having to take a sample and then run 4 or 5 different tubes, you'll be able to just run one tube with 40 colors in one single tube, and thereby saving on cost and running cases much quicker. It's also extremely valuable because it gives us an understanding of how to look at hematopoietic cell development in really, really deep phenotyping. So we're able to understand very rare subsets of cells and disease into different diseases. And this is really the future. I mean, if you follow any of these technologies over time, it's all about denser data and more multidimensionality, from, again, 4 color I wrote here to 6 color to 8 color, 10 color, 12 color is routine now. And then again, it's going to keep going on to more of the 30, 40 color. The reason that this data actually is quite accessible even today, and we're going to come back to this particular technology, is because of the development and the advancement in artificial intelligence. This artificial intelligence, the growth in artificial intelligence really has synergized perfectly with multidimensional data, and we're going to be able to just diagnose these complex cases even much easier. So that said, back to bpdcn. So bpdcn, we're going to cover a little bit of the pathogenesis and molecular insights. So bpdcn, the pathogenesis is that this arises in plasmacytoid dendritic cells, and there's aberrant NF-kappa B pathway activation and also type 1 interferon signaling. The genetic landscape is characteristic. You see TET2, ASXL1, and then spliceosome-related gene mutations, specifically ZRSR2 mutations in patients with bpdcn. ETV6 deletions are an early event in bpdcn. The prognosis and treatment is as follows. So it's typically poor with conventional chemotherapy, but there are newer therapies such as anti-CD123 therapy targeted treatments that are available. And there are clinical trials ongoing for BCL2 inhibitors, hypomethylating agents, and other types of approaches. Stem cell transplant is actually a good central therapy for bpdcn and can be curative if patients can tolerate it. So that said, we're now here in our outline for our talk, and we're going to now discuss Castleman disease, another rare and orphan disease. And again, it's good to come back to what is a rare and orphan disease. It's a disease where there are fewer than 1 in 200,000 to 2,000 individuals affected. And in the case of Castleman disease, the statistics vary, but it's somewhere between 10 to 30 patients per 1 million people are affected by Castleman disease. It was first described in 1950 by Benjamin Castleman, and most of us hematopathologists have seen, you know, several cases of Castleman disease, if not cases that look Castleman-like. It was originally called giant lymph node hyperplasia and angiofollicular lymph node hyperplasia. Since then, there's thousands, of cases of Castleman disease that have been described in the literature, and the amount of focus and research that's been going into Castleman disease has just exponentially expanded since the last decade. So from a pathologist perspective, when we look at Castleman disease, we think of it according to certain histopathologic kind of types. We look at Castleman disease and we think of the plasma cell variant, where you get lots of plasma cells, just like the name says. The Heinlein vascular variant, where histologically, when you look at a slide, you see lots of vessels that are hyalinized. And then the mixed variant, which essentially has features of hyaline vascular and also the plasma cell variant. Castleman disease, clinically and diagnostically, is separated out into two different main types. You can think of the unicentric type and the multicentric type. Unicentric type of Castleman disease has one site affected, whereas just like the name says, for multicentric Castleman disease, many different sites will be affected. In unicentric Castleman disease, we can separate them out based on the histopathologic type. And then in unicentric Castleman disease, one thing to note is that follicular dendritic cell sarcomas can arise out of unicentric Castleman disease and are something to be aware of and to look out for. Multicentric Castleman disease can be separated out into three main kind of types: HHV8 positive multicentric Castleman disease, idiopathic multicentric Castleman disease, and Poems type of Castleman disease. Again, with multicentric Castleman disease, there are those essentially three histopathologic types of a hypervascular, plasmacytic, and mixed variants that you may see when looking in the microscope. Over the years, because of our recognition and understanding of Castleman disease as far as histopathologically and what it looks like, and pathologists' interest in trying to describe the morphology, then in the last several years, a grading system for Castleman disease has arisen. A grading system that goes from grade 0 all the way to grade 3, looking at different types of features, looking at how regressed follicles look in Castleman disease, how much follicular dendritic cell proliferation, how vascular, how much hyperplasia of germinal centers, and how much plasmacytosis you see in Castleman disease, all with the attempts of actually putting a little bit more objectivity into an otherwise, gestalt kind of a feel to a disease. However, that said, the grading system for Castleman disease is tough. The grading can be subjective in some sense. And when we think about the grading of Castleman disease, where you have these different, 0 to 3 that I'm showing here, and you think of lymphomas, at least, it reminds us of a different disease, and that is follicular lymphoma. That's another disease that has a grading schema according to by ICC of grade 1 all the way to grade 3, and then grade 3, A and B. And interestingly, follicular lymphoma, there's many cases and has been studied for many years, and there have been attempts to objectivize, I put here, follicular lymphoma, where the grading of follicular lymphoma and identifying central blasts versus centrocytes has been difficult to do. But we're in an era now where there are things like digital pathology and computer algorithms that can be used to try to create objective criteria or objective data to identifying large central blasts versus smaller centrocytes. And I'm just showing a couple of figures from papers where there have been attempts in the last couple of years to actually utilize computational pathology and digital pathology to objectively identify grade 1, grade 2, grade 3, A and B follicular lymphomas. So you could imagine this might be a modality to use in other situations, like even Castleman disease. And I'm just bringing this up as something to watch out for. That said, I have said this word or mentioned digital pathology. And these digital pathology tools obviously require digital pathology. And so what is digital pathology? So digital pathology is essentially where we are looking at glass slides on a computer screen, and it requires several things. It requires a digital slide scanner to produce images from glass slides that you can then look on a computer screen. And it utilizes an IMS or an image management system to manage those images that are being produced by that digital slide scanner and then display them on your computer screen so that you can look at them. So most of us are now familiar with or aware of at least some of digital pathology, either using it in practice or again, just sort of peripherally aware of it. But why digital hematopathology? And I already told you that there's research work going on for grading and follicular lymphoma. There's a lot of practical reasons why digital hematopathology makes sense. It allows medical directors, MDs to work remotely, allows easy sharing of cases between various consultants all around the world. It's greater flexibility. People can work from home. It allows quicker access to your cases. If slides are coming off of the stainer and then loaded into a slide scanner into the IMS, you can immediately review them essentially. Cases can be archived very easily in a database and then pulled for tumor boards so that you're not looking around for glass slides all over your office. And then a key one, which we are going to spend some time talking about artificial intelligence, but the key one is that with digital hematopathology, digital pathology, with databases, with data warehouses, it facilitates, and all the data that hematopathologists we produce, pathologists we produce, it facilitates the development of artificial intelligence models. And so it's really a way to gather data to be able to utilize it to develop other tools. So this field is advancing very rapidly right now. Digital pathology is being utilized in immunohistochemistry at this point. It allows easy viewing of immunohistochemical stains and biomarkers where you can overlay multiple stains at once to be able to see if different areas are expressing different markers or not. It also allows hematopathologists to, again, quickly kind of order many stains. And the list of protein biomarkers in immunohistochemistry for hematopathologists is just growing immensely. And so digital pathology is actually helping to fuel this as well, because it's easier for us to understand what's going on in our different types of tumors, which cells are expressing what or not expressing what markers. In digital pathology, there are other ways that, and we're going to again come back to artificial intelligence, but how artificial intelligence is already being used in different areas of hematopathology. Some areas like bone marrow counting, where different algorithms are actually able to pull out different cells like myelocytes or blasts or eosinophils. And so this is something to watch out for into the future, to be able to use it in your own practice, or to be aware of it. So back to Castleman disease, though, we took a little bit of a detour talking about digital pathology. Castleman disease, again, separated out into unicentric and multicentric Castleman disease. There are different types of multicentric Castleman disease, and one to hone in on in particular, because there's been a lot of work done on it in the past few years, and a diagnostic system that has arisen, is idiopathic multicentric Castleman disease, which represents actually a half of the case or more of the cases of multicentric Castleman disease. So there are criteria for the diagnosis of idiopathic multicentric Castleman disease that have been published and are also included in the new WHO published by David Feigenbaum and also colleagues. And so the criteria, even though when you look at this table, it seems like quite a bit to pay attention to, but there's a way to kind of conceptually think of this. And then if you can't remember, go back to the WHO book or or look at the Blood article, both. So there's different criteria, major criteria and minor criteria. And the major criteria, the way I think about it is essentially looking if the histology looks like Castleman disease. That's one of the criteria. It has to look like Castleman disease by the pathologist's glass slide. And then also, and this is an obvious one, that for one of the major criteria, it has to be multicentric, so multiple sites involved. Minor criteria, the way to just generally kind of think about this is that there need to be laboratory values that support systemic dysfunction, because in idiopathic multicentric Castleman disease, there's sort of general systemic dysfunction for patients. And then there have to be other kind of there can be other clinical kind of findings like organomegaly, B-type symptoms, skin lesions, etc. And essentially what those are indicating is that there is a severity of the manifestation of this disease. Critical to the diagnosis of idiopathic multicentric Castleman disease is that you must exclude other diseases. And so other diseases that must be excluded are autoimmune diseases like lupus, lymphomas, plasma cell neoplasms, and more. What's interesting about idiopathic multicentric Castleman disease is, as you know, insofar as there's been so much study on it and research that's gone on in the last decade or so, we still don't absolutely know what idiopathic multicentric Castleman disease is. Is it some sort of an autoimmune disease? Does it have to do with a paraneoplastic phenotype? And so there's actually it's a cancer, or it's a systemic manifestation of a cancer, or is it infectious? And these haven't been totally worked out. There's some evidence for a neoplastic cell of origin, as well as some cases that have more of an autoimmune phenotype. Research into the infectious manifestation of idiopathic multicentric Castleman disease seem to point away from that. So where are we with our talk thus far? We're now still in the body of our talk, and we're on to another type of rare and orphan disease, pediatric type follicular lymphoma. And we're going to be brief here because there's much still to cover. But pediatric type follicular lymphoma is just like it says in its name, a follicular lymphoma that's seen in children and young adults. It's a rare disease. Generally, you'll see localized lymphadenopathy, usually in the head and neck region in patients. And it's a disease of just like adult kind of follicular lymphoma or classic follicular lymphoma. It's a disease of B cells. So pediatric type follicular lymphoma will be CD20 positive, and then also express germinal center markers like CD10 and BCL6 and HGAL. BCL2 protein is generally negative or very, very dim. And that's because there's no BCL2 or BCL6 gene translocation in follicular lymphoma, unlike the follicular lymphoma that's seen in adults and elderly patients. There are characteristic somatic mutations in the MAP kinase pathway, and then also point mutations in IRF8 like K66R that are quite characteristic of pediatric type follicular lymphoma. That said, in order to diagnose pediatric type follicular lymphoma, you need FISH, cytogenetics, or molecular studies, evaluation. And so what kind of technologies do you have to look out for? Since we said, you need cytogenetics and molecular evaluation, I just want to bring up some newer kind of advancements in cytogenetics and molecular that we should be paying attention to, and are going to be important more so into the future for all of our kind of diagnoses in hematopathology. Some which will be more upfront, and you'll see more frequently, like next generation sequencing. We're all quite aware of that. And then others where, such as AI-enabled karyotype analysis, which I'll show right now, which may be a little bit more behind the scenes unless you're actually running a cytogenetics lab. So we'll cover here now, and I'm trying to bring in some of newer technologies in cytogenetics, genetics, and genomics, but we're going to be quick because this is a time-limited presentation. And I do want to also cover a bit about artificial intelligence so we can talk a little bit about the advancements there. But in cytogenetic analysis, we can see already the application of artificial intelligence to making karyotyping much more efficient and accurate. And you can see what you're seeing is on the left side of here, manual karyotyping, versus on the right is artificial intelligence assisted karyotyping, which is able to pull out all of these different chromosomes and align them very easily for any technologist or pathologist to review. In genetics and genomics, technology has been advancing over the years, in particular in areas of high throughput sequencing or next generation sequencing, in the area of long read sequencing, and also in digital droplet PCR. And so those are three kind of areas that I'm going to briefly touch upon as far as being aware of newer technological advancements that are happening in those areas. High throughput sequencing, we're all familiar with by now, or should be. This is next generation sequencing. And there are different ways of doing next generation sequencing, from targeted sequencing to amplicon-based. And over time, the kind of advancements that we've been seeing in this technology is increased accuracy, increased effectiveness, new types of chemistry that, again, are leading us to more accurate, efficient sequencing, and also cost savings. And you can see those cost savings over time that are mapped here, and it'll just continue to go down, 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 as far as allowing us to also sequence many more patients and move towards more whole genome sequencing for any patients rather than targeted sequencing. So for high throughput sequencing, currently, it is used and can be used for DNA analysis, RNA analysis, and then also for epigenetics. These are different areas that are important for actually our classification systems, not just in hematopathology, but in other areas like in brain tumors as well. Epigenetics plays a key role, analysis or methylation analysis in being able to diagnose different types of brain tumors. So molecular phenotyping, what we've seen over the years is it's just continuing to increase. And so this is why it's very important to pay attention to this technology and know where it's headed. Long read sequencing is continuing to be adopted more and more so on a practical basis. And there are different ways of approaching this. Nanopore technology I'm showing here allows us to read, rather than short sequences that are, small in size and less than 1,000 base pairs. These are now very large, and you can see on the order of thousands of kbs. And what the importance of long read sequencing is that long read sequencing does really allow us to interrogate some of those structural abnormalities in more depth. And so we're going to see more and more of the adoption of that technology over time in hematopathology, in tandem with short read sequencing. Our bioinformatic technologies have advanced over time too. These tools have become easier to use for everyone, allowing us to call all kinds of different abnormalities, from indels, translocations, single nucleotide variants to CNVs. And again, as these technologies become easier and easier to use and they are more readily accessible, the impact and integration into diagnostics grows. And so remember, that's the theme here. So that's why when we're looking at technologies, pay attention to the magnitude of adoption, even on the research side, because it eventually trickles down or trickles through to practical diagnostics. And we see that have seen them over the years. Digital droplet PCR is also a fantastic technology that allows for high sensitivity and high specificity of identifying different types of variants. It's very robust. And at this point, you can target up to 6 or more, actually, molecular variants in a single reaction. And so it really allows the targeting of certain variants at a very low allele frequency. So very useful for things like minimal residual disease, so MRD. And you can imagine using it. And it is practically being used for identifying things like KIT mutations, JAK2, and then in the future, you can identify things like IRF8 mutations in pediatric type follicular lymphoma. So all of these technologies that I've been talking about, they will improve our diagnoses. And there's so many of them that are being currently used in hematopathology, from flow cytometry, cytogenetics, to molecular, to digital. But really, hematopathology is a multimodal type of discipline. And so with that kind of multimodal data, this brings us to the next pivotal type of technology for the future. Multimodal data lends itself so well to artificial intelligence, to computational analysis. And it's been incredible to see the field evolving just over the last 3 to 4 years. So first, in talking about artificial intelligence, we're just going to cover just a little bit of background on the current types of artificial intelligence, which, again, most folks should be somewhat familiar with at this point. Artificial intelligence, the definition of it is algorithms that learn and solve problems like humans. And there's different types of artificial intelligence in general. Applied artificial intelligence, which I think of as being practical artificial intelligence and how pathologists use it on a daily basis. That would be applied artificial intelligence, which is also a kind of a narrow or weak artificial intelligence, a focused artificial intelligence. Machine learning is any algorithm that learns from data. There's deep learning, generative artificial intelligence, which we're going to cover in the next couple of slides. Augmented intelligence, which is augmenting human intelligence using artificial intelligence, and then strong artificial general intelligence, which is something we're not going to cover today. So generative artificial intelligence has been getting quite a bit of buzz in the last few years, and it should, because this is a type of artificial intelligence that can create new content, new ideas. And really the foundation for really spurring this exponential growth in the last couple of years has been this transformer architecture that takes an input like words, is able to encode it, go through a decoder layer, and then create kind of new content. Take a sentence like "the dog is green," and then add another content and say "and blue," and predict how the sentence is going to continue on. So that kind of prediction or generation of additional new content idea lends itself really well to actually not just textual analysis, but also actually even visual data as well, and computer vision. There are certain types of other architectures and algorithms that are used in generative artificial intelligence for computer vision, though. So another one is generative adversarial network scans, and then also diffusion models, which are showed a little bit of what that architecture looks like here, for being able to take images and then analyze those images and create actually additional content. Hallucinations are something we should all be aware of in generative artificial intelligence, which again, generative artificial intelligence is artificial intelligence able to create something new, new ideas, new content. And one thing that is important terminology is hallucinations. So generative artificial intelligence can create ideas or content that actually is as much as it is literally a hallucination, is not real. And so one example that many of us are aware of is that if you ask things like ChatGPT to create, identify different references for in a review article, it may end up spitting out references that are seem factual, but are not real. And so this is an example of that here, because it is trying to predict the next sentence, the next word in a sentence. It tries to create it can create things that look humanistic, but are not actually factual. So another aspect of generative artificial intelligence, which is needs to be we need to be mindful of, is the fact that even though we humans have designed the architecture behind generative artificial intelligence, these transformer models, a lot of what's kind of going on within the algorithmic layers, it's a little bit uncertain on why it's giving the particular answers that it's outputting. And so there's an aspect of generative AI that's actually quite black box. And so around that, though, there are certain ways that we can try to make sure that there is less of a black box nature and uncertainty to what's going in and why an answer is coming out, by trying to increase the transparency around generative AI. And I'll talk about that a little bit later. But generative AI right now is, again, getting a lot of buzz and interest, and rightly so. It's not all just hype, because it can be used for algorithm generation. So you can use it to actually help you code. It's useful for actually genomic analysis even now, and that makes sense. Generative AI is very good at taking textual types of data, which genomic information, A, T, C, G, that's essentially like text. It's very good at taking that content and then predicting different outputs based on training sets. It's also very useful for workflow optimization as well in practical use. The other area where generative artificial intelligence, we're starting to see it pop up, is being able to, again, take multimodal, multidimensional data and aggregating it together and annotating reports. So report generation by artificial intelligence is an area of a lot of investigation, and some pathology institutes are even utilizing it now, where generative AI like ChatGPT can take in textual files, can take in flow cytometry information and reports, molecular data, bone marrow reports, and then aggregate all that information together and then create a final integrated diagnostic report that is actually correct and useful. Of course, a human is important to be involved in this process. And that brings us to some of our best artificial intelligence practices. As we go forward, it's really important to have transparency, safety, data security, robustness, and accountability with artificial intelligence. Because there is a positive, a bright side to artificial intelligence. And I think a lot of us are looking at artificial intelligence this way. It's going to improve patient care. It's going to give us better diagnoses and allow us to have improved testing. It can make our work easier by creating efficiencies, right? But on the flip side of it, there's also a negative aspect that we have to watch out for, because, like I talked about before, artificial intelligence, generative artificial intelligence, can hallucinate and create answers that are not actually correct and real. There can be a lot of uncertainty in those diagnoses. It can create more busy work in the end, because in any report that, let's say, artificial intelligence is generating, you may have to read through every single line in such detail that you can't even trust whether the noun is correct or whatnot. So it could create a lot more busy work. Artificial intelligence used in digital pathology too may end up flagging a lot of cases that are not, let's say, that aren't cancer, right? And then human has to go back and keep looking at these cases and trying to figure out why the algorithm called identified a neoplastic area when it actually isn't. Another potential dark side of or negative of artificial intelligence is you could say, well, pathologists are going to be replaced if you're looking at the negatives. But ultimately, we'll be able to do more work for our patients, and I think we're going to have to decide and have to really steer this ship going forward, taking responsibility for the future of pathology. Um, and we're going to have to take responsibility for our role in artificial intelligence because as much as those who are on the extreme side of how artificial intelligence is going to replace humans, I think those of us who are a little bit more practical and uh, in the weeds here recognize that we're going to have a human in the loop for many years to come in order to trust the kind of output that and diagnostic assistance that artificial intelligence is giving us. And so conclusions in the future. Technology will continue to drive hematopathology forward, and you've seen different instances of integration of these technologies. Multidimensional data is going to help us with our diagnostic accuracy and improve patient outcomes. And artificial intelligence will require adaptation and adoption of these new tools and methodologies. We can't be left behind. We have to take control of this ship. And then sensitive and cost-effective, and we all know this, user-friendly technologies are the ones that are going to replace older technologies. So that's always the theme. So thank you, and I can take any questions.
Dr. Ohgami, thanks for this very comprehensive overview on the innovations and orphan diseases in hematopathology. There are a few questions, Dr. Ohgami. One question that I see on YouTube is that what would you like to comment on the use of AI in flow cytometry?
Sorry, the use of AI in flow cytometry.
Yeah.
Yes, it's interesting that artificial intelligence, machine learning approaches haven't been adopted even sooner for in flow cytometry. I can say that there are different institutes right now that are using machine learning approaches to help triage flow cytometry cases to identify cases that are acute leukemias or cases that are more benign. And that kind of practical application of taking digitized kind of data, because flow cytometry data is digital, taking that data and then being able to identify, again, create operational efficiencies by identifying acute leukemias, and then organizing a pathologist's queue to put the most important ones up top and the lesser important ones at the bottom is something that the ARUP is already adopting, and we'll see more of that use in the operational side, I think, across different labs.
Thank you, Dr. Ohgami. Since there's another question on YouTube, I could see. So one of our viewers is asking, in regards to the uncertainty in AI generated diagnosis, how do you get around the black box to ascertain how the tool arrived at that conclusion?
Yeah, ultimately, it's impossible with these algorithms to get down into the nitty gritty and understand exactly why it gave the output it gave for every particular answer, because you'd have to, sort of tracking it is beyond us. But there is a way to make it a little bit less black box in the sense of understanding the larger architecture and the training set that was used, right? So that gets around to the black box and saying, look, we trained this convolutional neural net with these particular types of flow cytometry cases. This was the input, and these were the metrics that came out of the output, the number of cases that were called positive, negative, etc. And so that's a way for us as pathologists to address the black box nature of it, to put, as much as possible, put some information there. And as far as the beginning stage, the end stage, and a little bit of the architecture in the middle of saying, I know at least how many layers I did, how many epochs I was doing for a particular training set. So that's how you can have transparency.
Right. No, thank you. One more question I can see that what are the current limitations and challenges that you can see in implementing AI tools in hematopathology labs?
Yeah, so there are operational challenges and limit to implementing artificial intelligence in hematopathology labs. The operational side of it is you need to have a good IT operations in order to implement AI, right? Just in general. So there's if you don't have a general setup like that where you have an IT team that actually can handle and manage some of these aspects, then that's just a general issue. I think there's also an issue as far as implementation of whether pathologists within your group will want to utilize these artificial intelligence tools, right? So that's a challenge in and of itself. And getting buy-in just in general from your institution to trust these algorithms. So operationally, personnel-wise, or pathologist, I would call it wise, I would say that that's another aspect to work out. Then there's also the regulatory aspects of it, right? And being able to have the right type of experts within your institute that actually know how to, outside of IT, within your pathology, let's say pathologist team, understand how to implement it, right? And how to go through the whole validation and deployment of these tools. So it can be a challenge to adopt them, again, from the operational side, from the pathologist side, from having that expertise in general. But there's always help around. You can reach out to colleagues who have implemented these tools in the past, and then like ourselves, and go from there.
Yeah, no, thank you, Dr. Ohgami. You have already mentioned about examples, but there is another question that I could see that do you have some examples of specific AI models or algorithms that have been used maybe in the research setting or that have been published in detecting hematologic diseases or malignancy?
Yeah. So there are companies and vendors who have products out there right now that are used for identifying different types of cells in peripheral blood samples, peripheral blood smears, in bone marrow aspirate smears, etc., like Scopio and also Cellavision. Those are two examples of companies that have scanners and algorithms that are able to take peripheral blood smears, bone marrow aspirate smears, and identify neutrophils, blasts, monocytes, etc. So that's an area where there's already deployment.
Right. Someone was also asking about recent advances in Castleman disease. I think you already mentioned it, but someone was asking about, I think, the specific targeted therapy for multicentric Castleman disease. Do you want to say anything? Thank you.
Yep. There are targeted therapies. So Castleman disease, at least idiopathic multicentric Castleman disease, we do understand that there is in general kind of a cytokine storm, and as well as particular cytokines, interleukins, which are really key in the pathogenesis of idiopathic multicentric Castleman disease. And one of those interleukins is IL-6. And there is a targeted therapy against IL-6 that works in many cases of idiopathic multicentric Castleman disease. So anti-IL-6 therapy can be used. Although, and that actually has been adopted in the NCCN guidelines. Although some docs will use also rituximab as a therapy.
Thank you again, Dr. Ohgami. I see one more question about AI actually. How do you think that AI can assist in personalized treatment planning and prognostication in hematopathologic or hematologic malignancy?
Yeah. How can AI assist in prognostication
And treatment planning? Yeah.
and treatment planning? It's interesting. There are simple rudimentary forms of AI, essentially kind of decision trees that folks already use for trying to parse out the prognosis of certain patients. So, for instance, in MDS, right, that our approach to MDS requires, as far as prognostication, looking at cytopenias and blast counts and all kind of the rest. And so you can actually input a lot of that information into decision tree algorithms to output, what the prognosis is of patients. Beyond, sort of those kind of approaches with MDS, I did talk about how in flow cytometry, AI algorithms are used already currently for being able to identify whether patients have certain types of acute leukemia, et cetera. And that does immediately tailor into whether they should get certain therapies versus obviously somebody who has a chronic leukemia. If an algorithm is immediately identifying somebody who has an acute lymphoblastic leukemia, then you're going to have to treat that quite differently than someone that the algorithm calls out as far as having CLL. So I see AI algorithms as far as having impact on the diagnostic side, which ultimately really does impact the treatment therapies that patients get, for as far as, oh, this is CLL, this is ALL, this is AML.
Thank you again, Dr. Ohgami, for this excellent talk on covering so many different aspects on innovations that are relevant to hematopathology. And I think these are all the questions that I could find on YouTube and Facebook windows. And for our viewers, just to re-announce that this lecture is accredited by City of Hope for 1 CME credit. And if you wish to get the CME credit, so all you have to do is to register one time. This is free registration. And you can see the website here. And I have also texted the website on YouTube and Facebook chat windows. You can get it there. And of course, the CME credit is available only if during the live lecture, not after the lecture. So if you go to YouTube later, you will not get the CME credit. So for the US participants, you can text the code to this phone number, that is 626-314-7448. And international participants, you can also get the CME credit. You just have to email the code to this email address, that is cme@coh.org. And I have texted the CME code multiple times on both Facebook and YouTube. So you can send it and make sure that you have to send a feedback to City of Hope CME department, and you must submit it within 1 business day so that you can get the CME credit or the CME certificate. And thank you again, all our viewers, and all the viewers that joined from different countries. And I could see that you had Dr. Ohgami viewers who joined from Ghana, Mexico, Ecuador, Ethiopia, Turkey, Italy, India, and many joined from the United States as well. And for our viewers, thanks for your support. And if you like our lectures, please don't forget to follow Pathcast on different social media platforms like Facebook, YouTube channel, Instagram, as well as an X, so that you can stay updated with all the upcoming talks. And our next lecture is on dermatopathology. Actually, we are going to start a series of lectures on dermatopathology, and we will have our first one that would be an introduction. That's in September 24th, and our speaker would be Dr. Hadas Kapsky, who is an assistant clinical professor at UC Irvine in California. Hope to see you all at that time. And thank you again, Dr. Ohgami, for your time and for this brilliant lecture. Thanks a lot.
Thank you.
Thank you.